Hi, this is Dr. Shravan Kumar, DNB resident, Department of Anesthesia, Manipal Hospitals, Bangalore. As a healthcare provider, in order to tackle today's COVID 19 pandemic, it is imperative to understand the basics of virology. That is the purpose of this presentation. So, in order to start off, we need to define a virus. What exactly is a virus? A virus is an obligate intracellular parasite which relies on the host for energy containing DNA or RNA as a genome, with the morphology being either naked or enveloped, reproducing by component assembly. So, a virus is technically non-living structure and must encode all the required processes which are not provided by the cell. The virus must also have the capability of assembling itself. So these are the properties of a virus. A virus per se is something which contains a nucleic acid at its core of its structures. A DNA or an RNA component is the most important structure. It also has a structural protein, and following that, with the addition of nucleic acids or enzymes, this becomes a nucleocapsid. So a nucleic acid, a protein structure, and enzymes. These form a nucleocapsid, which is called as a naked virus if this is the completion of the whole structure. If along with this naked virion structure, it has an encapsulation with a glycoprotein and or a lipid bilayer, this becomes an enveloped virus. So this is a conceptual difference between a naked virus and an enveloped virus. A naked virus, as was told in the previous slide, has a nucleic acid and a structural protein, along with a few proteinaceous structures or enzymes. When this whole assembly is sitting inside an envelope, which is made of glycoproteins and lipids, this would be called as an enveloped virus. Now, based on the understanding of this structure, the properties and the virulence changes between the two types of viruses. So this is again a reinforcement of the same statement. A naked virus consists of nucleic acid, structural proteins, and nucleic acid binding proteins. This itself would be called as the naked virus. And what is the implication of this? It's a general assumption that naked viruses are not as protected as the enveloped viruses and hence the virulence. But that is not the case. Naked capsids are extremely rigid. They can withstand harsh environmental conditions and they have a very tough exterior. They are transmitted by fecal oral route and hence they can endure transmission in a sewage. They can withstand high temperatures, acids, proteases, detergents, and drying. They are generally released from the cells by lysis. They can spread easily by fomites, small droplets. They can survive dry surfaces and they can also survive the adverse conditions of the gut. Antibody, if the body is able to produce antibodies, that would be enough to contribute for immunoprotection. If the sewage is improperly treated, then the source of infection can be the tap water as well, or if there is any contact with the sewage. Based on the understanding of how a capsid assembles, we can have an idea about the antiviral drugs that can be manufactured or what is the pathogenesis of the naked capsid. A capsid is assembled from the structural component, which is a subunit, following which there is a protomere, a capsomere, a procapsid, and finally the capsid. That is what is represented here as a chronological sequence. Okay, so this is about capsid assembly. Now going over to the enveloped viruses, how this differs from that of naked viruses. Here, as it was already said before, there is a nucleocapsid structure. There is a membranous protection, which is lipid, protein, and glycoprotein. An enveloped virus is generally something which is transmitted via respiratory droplet mechanism. It also is spread by blood or tissue. By nature, enveloped viruses do not survive the harsh conditions of GIT, and they require an aqueous solution for transfer. So most of the droplet infections, the respiratory infections, are enveloped in nature, and the COVID-19 is no exception. It generally is transmitted by with the help of aqueous solutions. However, there are a few differences with COVID-19, but it's beyond the scope of my presentation. The enveloped viruses are environmentally labile. They are disrupted by acids, detergent, drying, and heat. This is in sharp contrast with that of naked viruses. They are released by cell budding and by cell lysis. And when they are released, they generally modify the structure of the cellular membrane. And in general, when they carry out the structure of the cell membrane, they also undergo partly some form of mutation because they are carrying the host cell machinery. So this is also different from that of a naked virus. The consequences of an enveloped virus would be that they are transferred via large droplets, secretions, organ transplants, blood transfusions. So a very good example of this would be the human immunodeficiency virus and the coronavirus. It does not need to be killed in order to spread. And here, unlike the naked viruses, not just the antibody, but cell-mediated immune response is also important for protection. This also elicits a hypersensitivity kind of a reaction. Another unique feature of the enveloped viruses is the presence of hemagglutinin glycoprotein, which is not just the hemagglutinin, the presence of unique glycoproteins, which are either used as the markers for the viruses, or they are used as the enzyme machinery to enter into a host cell. Like, for example, there is the VAP virus, which is a viral attachment protein. In the influenza virus, which is the H1N1, we have the H component and the N component, which is actually a VAP glycoprotein. The HA is the hemagglutinin glycoprotein. It binds to the erythrocytes, and the neuraminidase protein binds to the neuraminidase protein. Now, what is represented on the left-hand side is actually the spike protein, which is a viral glycoprotein with the N-linked carbohydrate. Now, exposure of a human or any mammalian body to this glycoprotein itself will elicit protective type of immunity. Now that we have a base understanding of what the difference is between naked viruses and the enveloped viruses, we can head into the basis for classification. Viruses are classified based on multiple systems, based on the structure itself, the biochemical nature, what diseases they spread, the means of transmission, the host cell, and the tropism. Based on the structure and the morphology, we have an array of viruses which can tell us. For example, the picornovirus basically translates to be a small RNA virus. The HIV virus itself can say that human immunodeficiency virus. So this is based on the overall morphology. So an HIV virus has a uniquely structural difference in comparison to that of the Ebola virus or the bacteriophage viruses. The rabies virus has a unique bullet-shaped structure. So I hope I'm clear with the point regarding the structural classification. The biochemical basis itself is based on the nucleic acid, which is either a DNA virus or an RNA virus. Based on the nature of the disease that they transmit, it can either be an encephalitis virus, a hepatitis virus, the HIV virus, the rabies virus. So I think this is also straightforward. And the means of transmission. Arboviruses basically means that the viruses which are spread by insects. And the host cells that are required, an animal, a plant, or a bacteria. And finally, to which organ it has a tropism to. The hepatitis virus has organ tropism to the liver. adenovirus, which has organ tropism to the lymphatic glands of the throat, the enterovirus, which means gut. So heading over to the actual subdivision, DNA viruses. This is a classification where it's further divided. The DNA viruses are divided into either enveloped or naked viruses. So enveloped viruses within the DNA viruses are pox viruses, herpes viruses, and hepatinovirus. Within the naked viruses of the DNA classification would be papilloma, polyoma, adenovirus, and parvovirus. So these are the different members within this within the DNA viruses. So within the pox viruses, we have smallpox and molluscum contagiosum. Within the herpes viruses, we have HSV1, 2, all the way up to 8. Adenoviruses, very straightforward. Papilloma virus, polyoma virus, JC virus, and BK virus. Parvoviruses, parvovirus 19. Hepatinovirus, which is hepatitis B virus. This is a busy slide, but the only thing which has to be taken home here is the size of the particles. They are all within nanometers, 300 by 200, and as we go down, the size of the structure is actually coming smaller and smaller. This is important in comparison between the DNA viruses, the RNA viruses, and overall size of our coronavirus, COVID-19 virus. RNA viruses are far more, and this is the division of the RNA viruses. What here is represented is the positive. The plus RNA basically means a positive stranded RNA virus. A minus basically means a negative stranded. A plus bar minus basically means that a positive or a negative stranded. And N is a naked, E is an enveloped. So purely positive stranded RNA viruses, which are naked, are picornoviruses, caliciviruses, hepavirus, and astroviruses. Enveloped viruses, positive stranded, is coronavirus, flavivirus, and togavirus. And negative stranded enveloped viruses are rhabdo, phyllo, orthoviruses, paramyxoviruses, bunyavirus, and arenaviruses. The ones which don't have a positive or a negative stranding are as follows: the reoviruses. Again, finally, an enveloped virus would be retrovirus. I think it's a spelling mistake here. It's a positive or a negative virus. So these are the different ones, of which the coronaviridae has COVID-19, the SARS virus, and the MERS virus. Even in this slide, what I've tried to focus is the size of the virion. Here, the coronaviruses are 80 to 130 nanometers in size. So this plays an important role in understanding the pathogenesis, because we need to understand how it enters into the type 2 pneumocyte or into the bloodstream. So coronaviruses they have a size of 6 to 7 into 10 to the power of 6 Daltons. That is the size of the virus. It's a positive stranded and a spherical virus. Now, finally, heading into the viral replication, there are totally 5 steps in the viral replication. The first step for any virus to enter would be the recognition. And recognition of the target cells. If it's an enveloped virus, it has those glycoproteins which are used for attachment to the target cells. Second stage would be the attachment of the virus. And the third stage would be the penetration within the cell. And finally, once it has penetrated inside, there is the uncoating phase, where it opens up its own cellular, the nucleic acid, and finally involves the messenger RNA, and it starts replicating its own genome. And some viruses have the property of using their genome only selectively, such that not only the whole genome is reproduced, but also part of the genome which contributes specifically to the protein structure or the overall pentameric or the overall capsid structure of the virus. And finally, once all of them are reproduced, they are eventually assembled into units, and they are either released from the cell in the form of lysis or by budding. As was discussed a few slides earlier, naked viruses generally leave the cell by lysis and they generally destroy the cell. Whereas when it is an enveloped virus, it does not necessarily destroy the virus. It can actually release with the help of budding and release, or purely by lysis mechanism. Enveloped viruses, they lyse the cell only in the presence of adverse conditions. They generally do not lyse lyse the host cell. Based on the understanding of the steps of replication, again, there is a basis for the understanding of the serology and the understanding of the antivirals. And finally, a brief slide on the COVID-19 replication. The spike protein that is present on the COVID-19, which is M protein, the S protein, it recognizes the ACE2, which is present on the type 2 pneumocyte. With the help of the ACE2, the spike protein binds onto the host cell and it is endocytosed with the help of the TMPRSS2 gene, and the virion is released. Once the virion is released, there is the translation of the genomic structure with the help of an RNA-dependent RNA polymerase. Because this is an RNA virus, it is RNA dependent. And the RNA polymerase is required for translation of not only the genome, but the structural proteins as well. Finally, when the whole translation procedure is completed, we have multiple RNA which are pure genome and also the viral protein structures. Finally, all of them are assembled with the help of the Golgi apparatus, and with the help of the exocytosis, there is budding and release of the new virion. Another important component which is to be understood is the viral genetics. Mutations, they generally occur spontaneously, and understanding viral genetics can help us in understanding the virology, the virulence of the viruses, and the nature of vaccines that have to be produced. So most of the mutations generally do not have any kind of effect. Most of the antiviral drug resistance is possible from the mutations. The RNA viruses, they generally do not have a mechanism of checking the errors. So the number of errors that occur in RNA viruses are significantly higher in comparison to DNA viruses, which means that the number of mutations within the coronavirus family also is significantly higher. Generally, if there is a mutation that has a significant change from the genomic structure of the parent, then it is considered as a lethal mutation, which is not sustainable with life. That causes the death of the viruses, and detecting these kind of mutations is significantly difficult. A deletion mutation means that a significant portion of the genome is removed from it. This changes the overall nature of the virus as well. If, let's say, a particular virulence protein is removed from the genome, then the possibility of the overall lethality of this virus is significantly lesser. There is another type of mutation called the plaque mutation, where a plaque of the viral genome is removed, and this also changes the overall nature of the virus. Because of plaque mutation, we can actually observe in clinical features the tissue tropism gets changed. A particular kind of virus which had tropism towards the lung tissue will change its tropism to a different tissue altogether. And another important thing is the attenuated mutant. This is what I was trying to describe in the deletion mutation. If a particular virulence protein is lost, then we have a significantly lesser virulent virus, which is extremely important when we are thinking of live vaccines. An attenuated mutant is extremely important here, which is the case in the rabies virus. However, this is in the phase of development, so it has not reached the market yet. And a conditional mutant, what it basically means is, in particular temperature or at a particular biological situation, the pH and temperature, the mutation occurs, and this can either mean that the virulence gets significantly increased or it can be lost. So these are the kinds of mutations that are present. And finally, the types of mutation that occur: the recombination, the reassortment, and complementation. Recombination basically means that within the host cell, the interaction between the viruses and the host cell basically causes a totally new type of virus which comes out. This can also play a major role in the virulence of the virus. Reassortment. Reassortment mutation means that there is a reassembly of the viral structure in such a way that whatever was intended to be a particular shape for the capsid is not what ends up finally happening. And finally, there is the complementation type of mutation, where let's say a particular virus has inability to find tropism towards a particular type of cell. We are not able to okay, we are not okay. Ruby has mentioned that it's almost 7:30. This is anyway my last slide. So in complementation, what ends up happening is there is a deficiency of a particular virus, and because it is deficient, it needs another input, either from the host cell or from a different virus, to complement the current genes. And only with that presence, it can actually obtain its virulence. So with this, I have come to the end of my presentation. So these are the different sources I had used. Predominantly, I had used the textbook of medical microbiology, and other data, other images I had obtained from these sources. Yeah. Thank you.